So today we're in AP Biology, Chapter 9. We're talking about patterns of inheritance or genetics. What we will be focusing on is the idea of how do we pass on our traits. And so one of the best examples of heredity is purebred dogs. They are extremely identical at their DNA, and that's because they've been selectively bred for one trait. So if you want long, floppy ears, you will only breed dogs that have the long, floppy ears. And you'll only breed that dog with other dogs with long, floppy ears, until eventually the genetics become the only alleles possible are those for long, floppy ears. And that's how we generate our purebred dogs. Whereas if you have a mutt or a dog of mixed breeds, then it has insanely large amounts of genetic variation, and that's because all kinds of different alleles are constantly being introduced. Now, when we talked in our previous chapter, our introduction to genetics, we talked about Gregor Mendel. And we talked about how Gregor Mendel kind of set the roots for science or genetics by doing some selective breeding experiments on his pea plants. And we talked about how he would cut off the stamen, which contains the male semen, and he would, using a paintbrush, he would transfer the traits from a flower of his choice directly onto a flower of his second choice, and then thus creating their offspring. So we talked about the pea generation having the parents. So here he took the carpel or the male parts off of a purple plant, and he combined them with the carpel, the stamen off of the male plant and combined it to the carpel of the white plant. And in doing that, what he ended up making was his F1 offspring. But unbeknownst to him at the time, what he found was he had only purple offspring. And because he did that, what ended up happening was he did this for several different traits. So he would specifically combine purple and white. He would specifically combine axial flower location, which is on the side, versus terminal, which is at the end. He would cross yellow and green. He would cross seed shapes, round versus wrinkled. And he wanted to see what would happen in the offspring. And by keeping very good and accurate tracking of this, he was able to develop some of his own laws. And so what he figured out is that for every single trait, you have two alleles, or two different versions of a gene. So, for example, the trait for flower color is the gene, but the two alleles you could have are purple or white. Now, from those two alleles, you're going to get one from your mother and one from your father. Therefore, they could potentially be different. Now, if the two alleles that you inherit are different, then there might be the one that is dominant. And the dominant one is the one that you're going to see expressed in a phenotype or those traits. Whereas on the other allele, that doesn't seem to show up in the appearance, but is still present in the genetics, is called the recessive allele. And so what Mendel was trying to figure out is that if you have two alleles, they'll have to separate when you make your gametes, your sperm or your egg, because you only donate one allele every time you do that. And so what he was saying is that if you have parents that have, let's say, two capital P alleles, well, then they're only going to be able to donate a capital P. And so you had the mother with the white, and she was only able to donate her lowercase or recessive P. But when you combine them, you're going to have one big P, one little p. Because you have the dominant P, it's going to look like the dominant color, which in this case is purple. However, it is possible when you combine a lowercase and a lowercase p that it is potentially able to make the recessive show up, because if there's no dominant present, then it will look like the recessive phenotype. Now, what is happening is that the homologous chromosomes have those alleles for each characteristic, and yet those alleles can be different, and they're going to be found at the same locus, so same location on the chromosome. So here we're looking at homologous chromosomes, excuse me, and we have our dominant P and our dominant P. These are homozygous. This means they're the same. Here, at a different locus, what we have is a lowercase a and a lowercase a. Again, these are the same, so we call them homozygous. And then we have on our last locus, we have a capital B and a lowercase b. Now this is telling us that we have a dominant allele and a recessive allele. Because we have one dominant and one recessive, we now say that this is a heterozygous for that particular trait. So there's a law of independent assortment, and we can actually see this happen by tracking two traits or characteristics at one time. And what we figured out is that we can see that the way that genes are separated is completely independent of what other genes are doing. And so here's an example. So what he looked at were two traits. One was for round versus wrinkled, and the other one was for green versus yellow. So he was looking to see, okay, if I have a homozygous dominant round, homozygous dominant for yellow, this is my P. I'm going to have a homozygous recessive for my wrinkled, and I'm going to have a homozygous recessive green trait for these. So they're both homozygous. This parent is dominant, this parent is recessive. So what's going to happen is they're going to create gametes. The only combination that this parent can create is by donating one R and one Y. Regardless of which R or which Y is selected, it's going to be a capital and O capital. This parent can only donate one R and one Y, but again, regardless of what combination they come up with, they're only going to create little r, little y. However, when they combine, what we're going to find is that this now becomes a heterozygous. It has a dominant and a recessive, a dominant and a recessive. And so it's heterozygous not only for seed color, but it's heterozygous for its round versus wrinkled. Now what we're going to do is say, okay, what if this one were to reproduce? Well, we have to look at all the various combinations that can be made in the gamete donating only one of the alleles. So this gamete can donate the first big R, and maybe it also donates the first big Y. So you're going to create the big R, big Y offspring. But again, maybe it creates the first big R, donates the first big R, but this time it donates the little y. And so you're going to create, where's my big R, little y? That should be a big R, little y. That one should be a big R, little y. And then what's going to happen is you can donate your little r and your little y, or you can donate your little r and the big Y. Now, what's happening is that by looking at this, possible combinations, I've done the FOIL method from math, and I think you guys are familiar with the FOIL method. First, outside, inside, last. Now, that is just for producing the sperm, but we have to cross this with the eggs. So again, we do the exact same combination, but this time for the eggs. And what we're going to find, so this one was done correctly. Big R, big Y. Little r, little y. There's our little r, little y. Our big R, little y. There's the outside. And then our little r, little y, which is our last for each allele. And we're going to cross this. Now, what we end up generating, if you look, is 9 of the potential offspring of 16 are going to be yellow with round. And those, again, are the dominant traits. 3 of the 16 are going to be green, but they're also going to be round shape. 3 are going to be yellow, which is our dominant color, but they're going to be wrinkled, which is our recessive. And then we're going to have our homozygous recessive. We have 1 out of 16. And it turns out that when Mendel did his experiments, this is what he found most of the time. 9 out of 16 were the dominant, homozygous dominant. 3 out of 16 were going to be heterozygous, which means they're going to have the dominant for one and then recessive for the other. And then we have our yellow wrinkled, and then we have our full homozygous recessive. This is important because it's showing us how to determine or use math to determine genetics. So here's a really good example of the independent assortment that happens. When you are mating heterozygous animals, what we can see is that this black lab here has a black coat and normal vision. So, and it turns out that black coat and normal vision are dominant. So we know that they have at least one dominant allele. We don't know what their second allele is, but we at least know that they have that. Whereas if you have a black lab that has the recessive blindness, then we already know 3 of the 4 alleles, because we know it has at least one dominant allele. And we know that in order to be blind, it has to be recessive, so both alleles have to be recessive. Now, what happens is that if you cross this with a chocolate lab, and we know chocolate is recessive. So again, this one has good vision. We know that in order to be chocolate, you have to be recessive for coat color, and you have to have at least one dominant allele in order to have normal vision. Again, here's our chocolate blind, which we know is completely recessive. Now, you can see here that what they've done is they've crossed 2 heterozygous offspring. What they find is that 9 times you're going to get the dominant black coat normal vision. 3 times you'll get the dominant black coat, but you'll get the recessive blindness. 3 times you'll get the recessive coat, but you'll get the dominant vision. And then 1 out of all of these 16 tries, what are you going to get? You're going to get the homozygous recessive chocolate and blind. So what geneticists do is they'll do a test cross to determine an unknown genotype. And what they're going to do is they're going to say, okay, here's what's happening. I have a black lab, but I don't know if it has a capital B or a lowercase b. And so what I'm going to do is I'm going to cross it with a known recessive lab, and I'm going to look and see. Now, if this black lab is homozygous dominant, then I know that all the offspring should turn out black, because no matter which B is donated, it's going to result in a dominant looking lab, which is black. But if this black lab over here has a recessive gene, it will still look black, but when it donates its gametes, it could donate the capital B, which would make black offspring, or it could donate the recessive B, which would create chocolate lab offspring. And so I know that by doing a test cross between these two, I can figure out what that other allele is. Because if I get all black, then I know that it's capital. But if I am able to produce a chocolate, I know that recessive gene had to have come from this parent over here. Now, what Mendel is doing is he created some rules that kind of tell us about probability. And probability is all about chance. Now, there are some rules that happen when you do the math of probability. There is the rule of multiplication. So if you're going to look at 2 independent events, what you're going to say is the chance that you'll inherit this coat, so black or brown hair coat, and you'll inherit blindness. What you need to do is you need to multiply to determine the true chance of having that event. Whereas if you're saying, I want to know what the rule is or what the chance is that I'll have a chocolate lab or I'll have a blind lab, what you need to do then is add them together. And what we find is that even though Mendel was looking at pea plants, so a common garden plant, he actually was creating traits or rules that applied to human inheritance. And so what we have started doing as geneticists or as scientists is we now create pedigrees. And a pedigree may sound like something for a dog, but really it applies to humans as well. And all it is simply a graphical representation of who has what genotype. So you give male squares and female circles, and you just simply shade in the ones that have a certain trait. And we can see if a trait is passed down in a family. And from that we can figure out, is it dominant, is it recessive, is it linked to what sex you are? You can figure all those things out. So here is an example of where we looked at being deaf. We can see that everybody as grandparents were normal. However, we did produce this offspring, Jonathan Lambert, which was deaf. And Jonathan went on to have a ton of kids, 2 of which were deaf. And so we know that it's a recessive trait. Now, there are tons of inherited human disorders that are controlled by one gene. Some of them are recessive and some of them are dominant. So some of the recessive ones are like being an albino. cystic fibrosis, phenylketonuria, sickle cell disease, Tay-Sachs disease. These are all recessive, and so it's extremely rare. Now, even the dominant disorders are extremely rare. And for one of them that's dominant is dwarfism, so being a dwarf or a little person. Some of the, like, Alzheimer's disease is a dominant trait. Huntington's disease is a dominant trait. In all honesty, you have to just memorize this table and figure out which ones are recessive and which ones are dominant. If you have a recessive disorder, which most human genetic disorders are, the odds of getting it are very small, 25%, and here's why. If you have a normal dad, you know that he has one dominant trait and then one recessive. The odds are that mom will also have one dominant and recessive. So mom looks normal and dad looks normal, or mom and dad can both hear. What happens is that they have to donate their alleles one at a time. And so if dad donates his dominant one, then his offspring will be normal or able to hear. But if dad produces a sperm that has his recessive allele in it, and that's used to make an offspring, then what can happen is that if mom does the same, then you end up producing the deaf offspring. However, just because you donate one recessive allele doesn't mean you necessarily are going to produce that offspring. In this case, if you look and see, mom donates the deaf recessive allele, but dad donates the dominant normal allele. And in this regard, we now create a carrier, an individual who looks normal but carries the recessive disorder. Some of the dominant disorders, like dwarfism, are still extremely rare, but they're around. Now, one of the things is that we have developed technology that allows us to look for these kind of disorders ahead of time. In this way, we can identify genetic carriers, and we can say, look, you're a carrier for this trait. Do you want to have the risk of producing an offspring that might have it if your partner also is a carrier? There are fetal testing that you can do. Amniocentesis or CVS are another one that can be done. And what they do is they withdraw fetal fluid, they isolate the DNA, and then they make a picture of it. And by taking a picture of it, they can look and see if there's any abnormalities in the DNA itself. They also do ultrasounds as a way of producing a picture, so we can look for physical abnormalities. And then at birth, they also do some simple tests in the hospital. They're required. They take a blood test to look and see, are there some simple genetic disorders? There is ethical considerations with this, because we have to say, well, what if you have a genetically defected person? Do you have a decision to make? Is that an issue? Is that a debate? Is having knowledge always the best route? And so it kind of has some ethical things. One of the things about Mendel's law is that there is a relationship between genotype and phenotype. And so Mendel applies to anything that is sexually reproducing. And so we have to remember that genotype does not often determine phenotype in such a simple way as dominant and recessive. And there's something called incomplete dominance, and this is an important thing. So here we have our homozygous dominant red flower and our homozygous recessive white flower. When you cross them, you're thinking, okay, I'm going to make big R, little r, and so it should look red. But that's not always the case. Sometimes you produce pink. And what happens is that heterozygous offspring is not one or the other. It's somewhere in the middle. So it's somewhere in the middle between red and white, therefore you get pink. So in this case, with incomplete dominance, you have a 25% chance of the dominant trait, a 25% of the recessive trait, and then a 50% chance of creating something that's incomplete or in the middle, and that's your heterozygous. One of the most health kind of factor related examples of this incomplete dominance is cholesterol. There are people who are normal with their low density lipoprotein receptors right here. And there are people that are homozygous recessive that have no receptors to remove cholesterol from their blood. However, there are people that are heterozygous and they have a little bit of the receptors. So they don't have as much as they should, they don't have completely missing. And so what happens is they have a mild form of the disease. They just have a little bit of the disease where they have too much cholesterol in their blood. Now, to make it even more complicated, there's a lot of times where there's more than two alleles that exist for a characteristic. It's not always just dominant and recessive, one trait. One example is blood. Our blood types in humans involve three alleles. So you can have allele A, allele B, and then allele O, or Yeah, O. Now, what happens is that because of that, we have to realize that it's not even A, B, and O, but it turns out that A and B are co-dominant, which means they're both going to show up, whereas O is recessive. And so, in order to be the blood type O, what happens is you make antibodies against A and B. You don't like A and B. If you're A, you're only making antibodies against B. If you're B, you're only making antibodies against A. If you're A and B, you don't make any antibodies. So what happens is that you can only receive blood from certain types of blood donors because your body would actually attack the wrong kind of blood. Now, one thing that happens is another thing that's called pleiotropy. Pleiotropy is where you have one gene, but it contains or controls all kinds of physical phenotypic characteristics. One of the best examples is sickle cell anemia. So if you have sickle cell, your blood cells are not round. Instead, they're kind of this weird crescent shape. Because of that, you get all of these traits, which leads to all of these complications. Your blood cells tend to fall apart. Because of that, you're weak, you're anemic, and you might get heart failure. Because of your sickle cell shaped blood, they oftentimes will clump or clog, okay? That can lead to heart failure, that can lead to pain, that can lead to brain damage, that can lead to kind of damage to your kidneys, your lungs, your liver, all of it. The other thing that happens because you have these weird clumping and kind of bad breaking down is you get accumulation of these cells in your spleen, which then damage your spleen. So all of these phenotypic effects are because of one gene, one trait. Now, one of the things that we always talk about is, or reference, is skin type. Our skin comes in various shades. We have the darkest dark to the lightest light. And because of that, what we know is that it's controlled by more than one gene. So it's called polygenic, more than one gene. And that creates this big continuum. So anytime you're looking at a trait, and the phenotype has a broad spectrum, then you know that the odds are pretty good that it's then going to be controlled by more than one gene, and we call it polygenic. Now, just because we talk about our DNA and our genes, we have to recognize that the environment will play a role as well, right? You can have genetic and environmental factors play a role. Your health might be determined by how many low-density lipoprotein receptors you have, but if you also eat a ton of cholesterol, you might have high cholesterol, regardless of how many receptors you have. If you avoid high cholesterol foods, you may not have high lipoprotein cholesterol in your blood, even if you're missing some receptors. So we have to recognize that genetics and in the environment play a role. We do have genetic testing to look at these kind of disease-causing alleles, and that's been a big factor and big step for cancer research. Now, what Mendel is talking about is how do we account for Mendel's kind of behavior or the chromosome behavior. Now, what happens is this all connects to meiosis, or when the cells are, or the DNA is dividing to produce sperm or egg. And so we have to make sure we look at the meiosis process. So here's our metaphase, so here's all of our DNA. Notice they're lining up and organizing, we're separating things out, and we're creating those gametes, those sperm and egg. We've talked extensively about mitosis and meiosis in our previous chapter. So make sure you revisit that and can apply that knowledge to the idea of how is it that we form those gametes, and then how does that relate to our genetics. Now, some genes tend to be linked together, and that means that you inherit them together. And when that happens, we have a pretty good idea that they're close together on the exact same chromosome. So, for example, one of these is the purple and red coloration and long versus round seeds. It turns out that they're located really close to each other, and so the odds are that if you have a purple color, you're also going to have a long pollen. And that holds true, and that gives us an indication that they're closely linked. Now, why? Why is it that some traits are inherited together and some are not when they're even on the same chromosome? And that's due to the crossing over that happens in meiosis. So what happens when you get your homologous pairs together and you form your tetrad for meiosis? These chromosomes are so closely interconnected that they might actually cross and exchange genetic information. So that when they separate into their individual chromosomes again, what you find is that you've now created some genetic diversity. Whereas before you just had big A, big B, and you just had little a, little b. But now what happens is you have the big A, little b, and the little a, big B combination that came out as a result of crossing over. One of the big things is that there was a famous scientist named Thomas Hunt Morgan, and what he did was he looked at crossing over in fruit flies. And what happens is he demonstrated that crossing over is less likely to occur between genes when they're very closely connected to each other on a chromosome versus when they're further apart on the chromosome. And so what happens is that scientists then use this idea to do a map of the genes and their location. And so what the idea is that the more often traits are recombining, then the further apart they must be. So the idea that the lowercase g and the lowercase l would be recombined or have crossing over occur between them is a direct reflection of how far apart they are. So the further apart you are, the more likely crossing over is to occur. And then we have to remember that there are some things that are directly linked to our sex, male or female. And so males have one X chromosome and one Y chromosome, which is very small. But they have one X chromosome, whereas females have two. And so when we look at it, sex is actually determined by the male. So if a male gives away an X chromosome, then it's going to be producing a female. If it gives away the Y, it's going to be producing a male. And that is because females are only capable of producing more female offspring. The Y chromosome has the genes that are responsible for the development of the testes. And the testes, they produce hormones that are responsible for controlling the male development. Whereas if you don't have the Y chromosome, you automatically develop ovaries. And so by default, the ovary development or female development is going to occur if there's the X chromosome. And it's only because the Y steps in and says, no, I think we'll develop testes, that we then develop a male characteristics. And so there are different systems that exist. So for example, in grasshoppers, it turns out that there are males and females, and it's just X and X or just X. Chickens have a completely different system. They have a Z and a W versus ZZ. So, and then honeybees are interesting because they actually have, the females will have more chromosomes than the males. Now, there are some sex-linked genes that do weird patterns of inheritance. And any gene that's found on the X chromosome is going to be sex-linked. Now, in Drosophila, it turns out that the white eye color is sex-linked. And so if you want to have a fly with white eyes, you're going to have to create the correct sex as well. So it turns out that by looking at the pattern of inheritance, we can determine whether a trait is sex-linked. And it seems to be that the trait is more likely to show up in a male if it's recessive, because think about it, males only have one X. So if they inherit the recessive allele from the one X, then there's nothing to cover it up. Whereas with a female, they have a chance of that second X being the dominant trait to then hide the recessive. And so it turns out that sex-linked disorders most often affect males. And that's because most sex-linked disorders are recessive, and therefore they're more likely to show up as the only allele in males, whereas females are more likely to be carriers of it, but hide it. And so one of the best examples is hemophilia. If we look at it, Queen Victoria was a carrier. She produced Alice, who was a carrier, and who produced Alexandra, who was a carrier. All three female were normal, but they carried the recessive allele. When Alexandra produced her offspring, Alexis, had hemophilia, and that's because both of them donated the trait. Now, what happens is that males who receive one X will have the disorder, whereas a female has to receive the disorder from both parents, and that's why we do genetic screening. That is our